Checking in. This is A&P 2, lecture test 1, video 8. And we're starting on page 19 of the note set. This is for Spotlight Figure 1716 on photoreception. So the first thing we're going to talk about is the dark adapted state. The dark adapted state is when you've been in the dark. And this works best after a half hour of complete and utter darkness. And I don't mean like dark, but there's like some lights through the blinds, or dark, but like the moon's out. I'm talking like deep in a cave, dark, 0 photons for half an hour. That's what I'm talking about. Okay, so dark, and I started a timer this time. I started it before because I'm always like, oh, I'll start it after, and then I start busy talking. And then I'm busy talking. So that happens. So in the dark, sodium ions continuously flow into the outer segment. And let me get my, come on, where'd you go? Come here. I'm supposed to be able to change this. Hi, it's not working. Arrows. I can't change. Oh, there it is. Pointer options? Pointer options. Let's go laser pointer. And then next, no. Black screen. Next. There we go. That was odd. Okay. Okay, so the sodium ions continuously flow into the outer segment. This is the outer segment, inner segment. Continuously flow into the outer segment from outside the cell. As you know, anytime you open sodium ion channels, sodium will rush in. Okay, so in the dark, the transmembrane potential of the outer segment here is a negative 40 millivolts. So back to the note set. Sodium ions continuously flow into the outer segment of the photoreceptor through something gated sodium ion channels, chemically gated sodium ion channels, kept open by cyclic GMP, which is just like cyclic AMP, but with a glutamine instead of an adenine. So G instead of A. Cyclic GMP. And that holds that gated sodium ion channel open. Okay. The constant flow of incoming sodium ions keeps the transmembrane potential around negative 40 millivolts. Lowercase m, capital V, volt was the dude's name. At negative 40 millivolts, rods and cones are constantly releasing the neurotransmitter glutamate, which is inhibitory, across the synapse to the inner segment. Oh, I'm sorry. The sodium goes onto the inner segment. The chemical, the glutamate, is released across the synapse to the bipolar cell. I misspoke. That means in the darkness, the rods and cones are constantly sending what message to the bipolar neurons? No light, 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 no light. So they're constantly sending an inhibitory message, screaming no light at the top of their lungs all the time in the dark. That's how you know that you're not seeing light. Okay. In the dark current, sodium ions flow freely into the outer segment, forward to the inner segment, and then out of the cell. Outer segment, inner segment, out of the cell. Okay, now let's add a photon to the mix. There we go. So now we've added a photon to the mix, so we're at number 1 in the picture. And at number 1, opsin is activated. So the photon, there's the rhodopsin, is the whole thing is an opsin, and then this little key shaped here thing is the rhodopsin and the opsin. Yeah. And the opsin is activated, and this retinal is changed from an 11 cis form, the bent form, to the 11 trans straight form. So the photon, number 1, activates the opsin, and the activated opsin changes the shape of the retinal. Okay. Next. Oh, yay, it worked. Number 2, the opsin, activated opsin, activates transducin. And the transducin activates the phosphodiesterase, PDE. So photon activates the opsin, changes the shape of the retinal, and the activated opsin activates transducin. The activated transducin activates the PDE. Next, what the heck does a PDE do? The PDE is an enzyme that breaks down the cyclic GMP. So number 3, the cyclic GMP levels decline, they lower, and the gated sodium channels close because we no longer have the cyclic GMP holding them open because the PDE has broken them down from cyclic GMP to regular GMP, and regular GMP does not hold the sodium ion channels open. Only cyclic GMP can do that because in the biochemistry, it's all about shape, and we've changed the shape. Okay. So why did the sodium ion channels close? Yeah, I said it right. Why did the sodium ion channels close? Because the cyclic GMP that held them open was broken down by PDE. And the PDE was activated by the transducin. The transducin was activated by the activated opsin. And what activated the opsin? A photon. So this is all from I see the light. Okay. Number next. 4, I think. There we go, 4. So the dark current is reduced and the rate of inhibitory neurotransmitter declines. So because we're no longer allowing sodium ions, positive sodium ions, to rush into the outer segment depolarizes, hyperpolarizes from its resting transmembrane potential of -40 millivolts to here we show a -70 millivolts. Now we still have sodium ion leaving the inner segment, and this change in the transmembrane potential of the photoreceptor decreases the rate of inhibitory glutamate transmission across the synaptic cleft. So basically, the photoreceptors are all no light, no light, no light, no light, no light, and then a photon comes along and they stop saying no light. So again, with the double negatives. So we've got no light messages, and that's how we know we just saw something. And that's it. Super complicated, but kind of simple. So it's a little of both. And then when we decrease that message, then the bipolar cell sends the message on to the ganglion cell that we have seen the light. Okay. So after that is bleaching. The rhodopsin molecule has to be broken down. Let's see if I can move this. Can I move this? No, I can't move it. Into the retinal and the opsin. So because the photon changed the shape, we actually have to tear the retinal out of the opsin, burn an ATP to change it back, reinsert it into the opsin before that photoreceptor, that visual pigment, can see again. So it's this whole big complicated thing called bleaching. And when you don't have enough retinal, which is vitamin A, to help redo the bleaching, then you get night blindness. And the term for night blindness is nyctalopia, and it results from a deficiency in vitamin A. Now, this difference between, oh, we skipped why is bleaching not normally noticeable. Normally, because your eyes are always moving the image on the retina, like we discussed with the plus and the circle, the optic disc blind spots when we were trying to find those. It's the same thing as you're constantly moving a little bit on the fovea, moving the image around on the fovea. And so when you're light adapted, the rate of photons coming in and the rates of bleaching are matched. But especially in night blindness where like it's dark and then you see like headlights and it kind of takes a second before you can see again, that's because nyctalopia, not quite enough vitamin A to remake those visual pigments quickly. So there is a difference between natural vitamin A and synthetic vitamin A, especially in pregnancy. So you need a certain amount of vitamin A for your eyeballs to keep the bleaching and the reforming of the visual pigments going. And this, when there's not enough, it's first distinguishable in the dark. So that's why it's night blindness. Now, natural vitamin A and synthetic vitamin A will both work for your normal everyday night blindness. You can overdose on vitamin A, more easily synthetic vitamin A. Natural vitamin A you can also overdose in. There's lots of vitamin A in livers, and especially in polar bear livers. There's so much vitamin A in polar bear livers that it can kill you. Yeah, so don't eat polar bear liver ever, ever. Okay. Now, when you're pregnant, you need a certain amount of vitamin A in your diet. I think it's 10,000 IUs or some such in your diet in order for baby to have enough vitamin A to make these retinals to have functional visual pigments when they're born. Okay. But too much vitamin A, especially too much synthetic vitamin A, can hurt babies' eyeballs and make them not able to see. Now, too little vitamin A and it hurts their eyeballs and they're not able to see. And too much synthetic vitamin A and it hurts their eyeballs and they're not able to see. So there is a range of vitamin A that is safe. And when you're taking both prenatal vitamins and eating vitamin enriched foods like white flour, white breads, and those kinds of things, then instead of some breads, some flours aren't enriched. So if you're having cereal and enriched bread products and taking prenatals, then double check your vitamin A levels every day. When I was pregnant, I read the labels on everything because that's what you do, part of it. So, but bleaching, you have to, since you've changed it from the 11 cis form to the 11 trans form, you have to pull the retinal out of the opsin, burn an ATP to put it back so that you can see again. Bleaching, fixing the rhodopsin, which is the visual pigment. Okay, that's page 19. Let's see what page 20 has to say. Page 20 says the dark adapted state, and I did mention this briefly. The dark adapted state is after 30 minutes in complete and utter darkness, as we mentioned. So this is when the visual system is most sensitive, all the visual pigments are reformed, fully receptive, and after 30 minutes in the dark, also your eyes will be dilated. The light adapted state, as I mentioned, the rate of bleaching equals the rate at which the pigments reform. It is 25,000 times less sensitive than the dark adapted state. And the pupillary muscles affect visual sensitivity. When you're dilated, you're getting 30 times more light entering the eyeballs than when they're not dilated. And the sensitivity of the entire system between being light adapted or dark adapted versus dilated or pupillary constriction, the sensitivity of the entire system changes of a factor of about 1,000,000 with an M. That's a lot. That's a whole lot. So, next. Oh, look, I had all that typed in. There you go. Retinitis pigmentosa is I missed my notes. It must be in my office because I've gone through my whole house. Retinitis pigmentosa was a progressive disease where the visual pigments degrade, but we don't know why. That's what I've got off the top of my head. Double check that. Color vision. What do we interpret as the color white? All the cones and the rods together is white. Black is no photons. That's black. And color blindness. Let me flip back to color blindness. I have the technical terminology written out here somewhere. Ooh, it's thundering here. I don't know if you can hear it. Okay. Color blindness is the inability to distinguish certain colors. The most common cause of color blindness is red-green, where you, I think, can't see red. Either don't have red cones, or the red cones, the rhodopsin isn't right. Color blindness is more common in males than females. In men, 10% of men have some form of color blindness, while 0.67% of women have some form of color blindness. 1 in every 300,000 people is totally colorblind. I do have a friend that's totally colorblind. And in fact, you know the Bob Ross videos where he like paints and happy little trees and happy little accidents? He did a video once. They're all on YouTube. You should totally watch them. They're all on YouTube. And he did a video once because he had a friend that was totally colorblind that told him that he couldn't paint because he was colorblind. And so Bob Ross did an episode of his painting show totally in black and gray. And it was amazing. One time my daughter decided last January to watch a Bob Ross video and paint a little scene, and she still has it, and it's incredible. So I'm not an artist. My husband is an artist. She did not get that from me. So she's amazing. Okay. She got my music and his art. How is that possible? Okay, so did we answer why color blindness is more common in males than females? No, not yet. Okay. Punnett squares. We're going to Punnett square now. Whiteboard. Actually, let's see the blackboard. Ooh. Okay. That's not helpful. Pen? What? I'm not sure what's happening. Uh, uh, That was supposed to be a smile, but it's clearly a meh. Okay. Ooh, color blindness in men, not women. Okay. This, my friends, is a Punnett square. So in Punnett squares, and I'm not teaching you all the terminology for this, we're going to do just the basic concepts as far as they are, as much as we need to know to do these as a logic puzzle. When we get to test 6 and we do genetics, we will absolutely learn all of the terms for this. In the meantime, basic concepts. Okay, so Papa's genes go down the side. So let's do the sex chromosomes. Sperm donors have an X and a Y. Mama's genes go across the top. So your egg donor has X's and X's, and that's it. So half of Papa's sperm that he makes will have X's. And half of Papa's sperm will have Y's. And half of Mama's eggs will have X's, and half of Mama's eggs will have X's, generally speaking. Now there are variations. You can have XXX, is that Weber's? I think XXY is Turner's. You can have fragile X, you can have all kinds of in between XX and XY. Although for many people with differences in the sex chromosomes that usually will cause sterility and they won't be able to have children, not always. Like people with XYY, with the extra Y chromosome, tend to be more aggressive and sterile. And so there's lots of variation, including now usually you would say like, oh, XX is genetically female, XY is genetically male, although it's simply not that simple because the gene on the Y chromosome can actually break off and attach to the X. So you can be genetically XX, where the genes look female, the chromosomes look female, but have the gene on the X, which causes maleness. And you can be XY, genetically male, but not have the gene that causes maleness. So you can be genetically XX female, but physically male, genetically XY male, but physically female. And there are lots of different genetic and physical combinations that can cause an in between. So there's society desperately wants, especially the people who aren't terribly bright, for things to be very simple and for there to be either male or female. And that would be simpler. I agree. But reality states that's simply not the case. There is a large proportion of the population that is intersex, that's in between male and female with some male, some female characteristics. There are lots of different variations. Um, we'll talk about this more when we get into the genetics. So basic concepts here. X is across the top, X, mama's across the top, papa's genes go down the sides of the Punnett squares, and male versus female is not as simple as society wants you to believe. Okay. All right. So what happens with the color blindness is it's on the X chromosome. So if your egg donor has normal color vision, then that will be indicated with an X with a capital C, a subscript capital C. And for an X with the color blind vision, that'll be shown as an X with a superscript, not subscript, superscript, lowercase c. So what was our question? Oh, why is color blindness more common in males than females? Because if, say, your mama is a carrier, so she has normal color vision, because if you have, as a woman, the normal color vision genes, then you make the normal color vision. But can carry the colorblind gene. So let's say Papa has normal color vision and is male. So you've got X's with normal color vision here, and an X with a normal color vision from Papa here, and a Y. And then here you have an X with a normal color vision from Papa, and an X with a colorblind color vision from Mama. So you can see that the daughters, all X's is daughters generally, normally but not always, that the daughters will have normal color vision and then normal color vision but carry the colorblind gene. Now what about this son? The son does not get an X from Papa, because sons never get X's from Papas, right? They only get Y's from Papa. So this boy gets the color blind gene from the carrier mama. So the sons, there's a 50% chance of the normal color vision son, assuming they're sons, and a 50% chance of the color blind son, person with testes. Either way. Okay, so that's why. Because what would we have to do to make a color-blind daughter? So we would have to have, okay, that part we know. To make a color-blind daughter, she would need a color-blind gene from the mama, and she would need a color-blind gene from the papa. So if this mama were a carrier, and Papa were colorblind, then they could have colorblind daughters. If Mama were colorblind and Papa were colorblind, then of course all of the kids would be colorblind. So Punnett squares are what biologists do on first dates. True story. Okay, heterosexual biologists do on first dates. Yeah. Okay, so that's, how do we, oh, there we go. Excellent. I'm not sure I like the black because I couldn't see where the edges were. I think next time I'll go back to doing the whiteboard. All right, so the dark adapted state, we already saw this one. We were here, no, we already had this conversation. Photons of all wavelengths bounce off objects. Rods are stimulated, but not the cones for white. And then the objects absorb black. The objects absorb all the photons. And then color blindness, red, green is most common and more common in men. How are we on our timer? We have 15 seconds left. How's that for time? Okay, we're gonna finish this page. Next page. I hear rustling in the background. All right, so the visual cortex is the part of, yep, there's our timer. Good, good. The part of the occipital lobe that receives the photoreception. The association areas will, of course, process that. The visual pathway begins at the photoreceptors, synapse from the photoreceptor to the bipolar cell, and the bipolar cell to the ganglion cell, and ends here at the visual cortex. Do we have depth perception on the next page? Oh, we do. So depth perception, and the picture's behind me, sorry. So the depth perception is you compare the distance from the two eyeballs. So you're kind of 3D kind of triangulating the information. I had a friend in high school who had a glass eye, and so she had no depth perception, but she had a car. And and she said we could go anywhere in town after school except downtown, because in downtown you had to parallel park, and parallel park you have to have depth perception. Now, I've had people tell me since that they don't have depth perception and they can parallel park. And I do have depth perception, but I'm still not great at parallel parking. But I haven't lived in a downtown where I've needed to parallel park for decades. So it's probably just a lack of practice. Anyway, the field of vision is what you can see. So, you can do the little thing with your hands where you find the edges of your field of vision. See it. Can't see it. That's your field of vision. And of course, do the, there we go, field of vision. And then your circadian rhythm is the day-night cycle, affects other metabolic processes, including the reproductive tract, So that's interesting. And then let's start the ear ever so briefly, just to finish off page 20. Oh no, yeah, equilibrium. Equilibrium is your balance. It's gravity, linear acceleration, and rotation. And hearing enables us to detect and interpret sound waves. Receptors for both equilibrium and balance is the hair cell, and they are mechanoreceptors. And the auditory cortex, excuse me, is right here in the temporal lobe. Water. Now I have a tickle, sorry. Mason jars for the win. We're a mason jar family. I wasn't raised on a farm, but both of my parents were raised on farms. And I'm from the Midwest, raised by people who were raised by farmers. So, mason jars. It's all about the mason jars. And of course, the auditory cortex receives the information of the sounds you hear, and they are processed by the auditory association areas. Boom. And then we start in the anatomy of the ear on page 20. So we did page 19 and page 20, and we have 21, page 21, page 22, page 23, and page 24. So we have probably 2 videos left. Yay. All right. And I don't want to do any updates about this semester specifically, because I want these videos to be more evergreen and consistently useful, and not all about test dates. So, but check your test dates. And there's always this lab material that we cover in the lecture on, like, the eyes and the ears and the taste buds and all of that is on the lecture test, because even though it's lab material, we did cover it in lecture. So don't wait till after the lecture test to study lab. That is evergreen, always and forevermore. Okay, so this has been a video lecture for A&P 2, lecture test 1, video 8, covering pages 19 and 20, and our first introduction to the Punnett square. That's right. Yeah. All right. So thank you as always for tuning in. The education must go on. Dr. J out after I find the command control escape. Okay, command control escape. Thanks, y'all.